As the title slide here promises, I'm going to talk a little bit about computer-designed organisms. This is work primarily by my PhD student, Sam Kriegman, here at the University of Vermont, and work together with our two biology colleagues, Doug Blackiston and Michael Levin, at Tufts University. Fabio mentioned Daniel Dennett, who's at the same university, so it's a small academic world here. Before I talk about computer-designed organisms, however, I wanted to take a step back and situate CDOs in the larger landscape of artificial intelligence and robotics, because on the surface it may not look like an AI and robotics enterprise, but I hope to convince you that it is. And I'm going to start by giving my opinion of the landscape of AI and robotics and where it is and where it may go in future. And in order to describe this landscape, I'm going to use a geometric metaphor. So imagine we have this this single dimension here, which is performance. And you can imagine any given machine learning algorithm as a line segment that's embedded in this dimension. Points within that line segment might correspond to agents being trained by that machine learning algorithm. And obviously points that lie further to the right are more performant or better at performing whatever task we're training them to do. And as most of you know, throughout the vast majority of the 50-year history of AI, most of our learning algorithms and robots didn't basically do much better than chance. So they hugged the left side or the origin of this single dimension. But thanks to convolutional neural networks and the deep learning revolution and the big data revolution, we're now able to create machine learning algorithms that are line segments that hug the right-hand side of this dimension and can train agents that become as good as, if not better than, human beings at recognizing patterns in data. If we add a second dimension, however, we can now think about agents performing different tasks. So now we have a two-dimensional space, and you can now imagine embedding line segments in this two-dimensional space, where again the line segment corresponds to a machine learning algorithm, and points along that line segment correspond to agents that are becoming better and better at the task we give them. They're moving from left to right in this two-dimensional space. And potentially, we may be training them to perform more tasks over the time period of training, which means we're moving up and to the right. So here I've just embedded some arbitrary tasks, and I've sort of organized them in order of difficulty, at least from the point of view of us trying to train machines to perform those tasks. We could argue about the order that these should be placed. It's not really important for our purposes. Just remember, this is a cartoon and a metaphor to guide our thinking. Obviously, we've now been able to train agents that are very good at discrete tasks where solutions are discrete, like checkers, chess, and Go. More continuous problems like computer vision and NLP and protein folding, which was very popular over the last few weeks. These are more continuous problems, more difficult, but we definitely are making progress on these tasks. What we're making less progress on, as I mentioned, is this line segment that moves up and to the right, training agents that become better and better at a growing number of tasks. This is still an open problem in machine learning. However, I think Fabio mentioned this as well. There are certain people within the field that believe that just with larger neural networks, with more data, with more computing clusters, with more carbon, with more energy, we will eventually be able to push our agents towards, if not reach, artificial general intelligence. And at the moment, that's just a belief. Some people believe in it, some people don't. If you add a third dimension, which is the one that we are most interested in, and Fabio, I think he showed this actual slide in his presentation. If we add embodiment as a third dimension, we can think embodiment is a very controversial term. Embodiment means a lot of different things to different people, but generally speaking, you can think of greater embodiment as an agent that has a more complex body. It has more mechanical degrees of freedom, more sensors, more motors, more ways of moving in the world, more ways of becoming damaged, more ways of interacting with the environment, and so on. So towards the back of this cube, I placed the complex Atlas humanoid robot. Roomba here is closer to the front square of the cube. Roomba is embodied, but it's embodied less than Atlas, and a naked neural network that is waiting for images to be supplied to it for pattern recognition is not embodied at all. So this third dimension is also a reminder that embodiment is not an all or nothing property. We can imagine agents that are more or less embodied. So now you can imagine other kinds of machine learning algorithms that lie along what's called the grand diagonal. They're moving along all three dimensions simultaneously. And again, I'm trying to represent this with a cartoon. So if we imagine points along this grand diagonal, those points correspond to agents that are being trained to become better and better, moving from left to right. They're becoming more and more general. They're able to tackle a greater number of challenges. But they are also, over the training regimen, becoming more and more complexly and interestingly enmeshed in the interactions with the environment. They're becoming more embodied. Myself and some others, but still a minority in the field of artificial intelligence and robotics, believe that the better path towards AGI, and again, whether we'll ever reach it is sort of a philosophical, not a scientific discussion. A better way to make progress towards useful, intelligent, and general machines is to allow the machine learning algorithm to alter their morphology during this process. Fabio also showed you a version of this cube where you have lots of curved segments branching and suffusing through this space. I think one of the things I like about this way of thinking about AI and robotics suggests that there are many different paths, literally in this cube, that you can follow as an AI or robotics researcher to try and make progress in the field. Fabio also showed this example, sort of state-of-the-art in robotics from Google. This is what's known as an arm farm. So we have 7 arms here, which were trained over 4 months to manipulate objects in the tray in front of them. They trained a 1.2 million parameter neural network, and at the end, these arms were able to successfully grasp unfamiliar objects in the tray in front of them. But if you think about the machine learning algorithm that trained these arms, that machine learning algorithm corresponds to a line segment that's trapped in a horizontal cut through this cube. The arms themselves, they all have the same morphology. Their morphology did not change over the 4 months. So this line segment does not move from the front towards the back of the cube. They were given the same task, not more tasks. So they're not moving from the bottom to the top of the cube. They're simply trying to train neural networks that move from the left to the right. So what happens when we relax this assumption that the arms or whatever the robots are that we're training have the same morphology over time? This is actually an old idea. It goes all the way back to the early 1990s and work by the computer graphics researcher Carl Sims. Back in the early 90s, Carl created his own physics engine that you see here. He then wrote an evolutionary algorithm that evolves populations of virtual creatures, like you see here, creatures that move faster. The evolutionary algorithm makes randomly modified copies of those fast-moving creatures, and those randomly modified copies replace slower-moving creatures. So again, a very simple version of evolution, but it produced these very interesting solutions to interacting with the environment. This was definitely a high watermark in the field of AI. It was very exciting at the time, and for many of us, it is still very exciting. 6 years later, Hod Lipson and Jordan Pollack reported in Nature a version of Carl Sims's algorithm, which you see in the top panels. But they used what was then a new technology called 3D printers to print the plastic parts of these evolved creatures, and then to manually snap in the motors and the electronics to allow these evolved virtual creatures to cross the reality gap. And as you can see in this video, many of these evolved creatures that were able to actually exhibit their evolved behavior in the physical world after only experiencing the virtual world during evolution. Moving forward in time, Nick Cheney, Rob McCurdy, Jeff Clune, and Hod Lipson again adapted this idea of evolving virtual creatures in simulation to now a new simulation which allows for the simulation of soft-bodied agents. So I'll let this video run as I explain what you're seeing here. You'll see the simulation in the top, and as you can see, we're now simulating soft-bodied robots using a finite element method, where the elements themselves are voxels or 3D pixels, little cubes. So what the evolutionary algorithm does in this case is each piece of DNA in the population places voxels within this empty cube at the beginning, and then it also paints different colors onto these voxels, where the color dictated by the DNA represents material properties of this soft-bodied agent. You'll notice that many of these agents are mostly made up of red and green voxels. Red and green voxels here correspond to the soft robot equivalent of muscle. So there is an open loop controller that is expanding and contracting the volume of the voxels. And red voxels and green voxels always expand and contract in antiphase to one another. This is like agonist or antagonistic muscle groups in mammals. There are two other voxel types, and you'll see a few of them sprinkled here and there throughout these evolved creatures. There's light blue voxels, which correspond to passive soft material. This is the soft robot equivalent of fat. And I don't know if there's any examples here, but evolution can also make use of dark blue voxels, which correspond to passive rigid material, which is the soft robot equivalent of bone. You'll notice I'm showing you here snapshots of the best or most fit robot in the population at any given time. So at any point in time here, you're seeing one individual that is extracted from a population of robots, all of which have different 3D shapes and different material properties. Lucas asks a question, which is, could we use the self-reconfiguring systems to evolve creatures without the need to rely on imperfect virtual simulations? That's a great question. So this has been an idea for a long time, which is that maybe if we make improvements to self-reconfiguring systems or modular robotics, we could do away with simulation altogether. And I think the jury is still out on that, Lucas. There's theoretically no reason why we couldn't do that. I think it's just a matter of building the hardware and seeing. So good question. Please do feel free to type questions into chat as we go. Okay, so I just going to spend a few minutes to dive down into the technical details for those that are interested, what's going on inside the evolutionary algorithm. Well, in this project and the computer designed organisms project that I'm going to show you in a moment, the DNA that describes the robot is encoded as a particular kind of neural network known as a compositional pattern producing network, or cppn. These were reported by Ken Stanley back in 2007. For those that are interested in robotics and generative systems and evolutionary systems, it's definitely worth your time and effort to go read through the CPPN paper and understand how CPPNs work. They're a very popular tool in our field. I'll give you just a taste about how they work. Here's a very simple CPPN that has two input neurons, X and Y, and one output neuron, G. There is a single synapse that connects X to the output neuron G, no synapse connecting Y to G. Imagine we have a two-dimensional space and we visit a series of points distributed uniformly throughout that space. When we visit each point in the space, we take the X and Y coordinate of that point, feed those coordinates into the input layer, propagate those two values down through the neural network, and the value arriving at G, we're going to interpret that value as the amount of gray that we're going to paint onto that point at that location. We then go to the next point, take its 2D coordinates, propagate them through the same network again, get a new value of G, paint that pixel, and so on. And what results, if we do this in a two-dimensional system, as you can see here, as we move from left to right, the values of the points, the X coordinates increase, which means we paint more G. We'll assume that this synapse has a weight of 1. So the greater the X, the greater the G. Very, very simple. You'll notice immediately, however, that this neural network results in a gradient of pattern being produced from left to right. Imagine we have a population of these networks with different synapses and different synaptic weights connecting the input layer to the output layer. Different CPPNs are going to paint different patterns. So imagine in the same population there is another neural network that has the same cognitive architecture, two input neurons, one output neuron, but it has different synaptic weights. So we can imagine the synapse from X to G is either missing or has a value of 0. And Y has a positive synaptic connection to G. In this case, as Y increases, and I've drawn it here with Y going from the top to the bottom, we paint more G into the space. We can also modify the activation functions inside the neurons, like for example, taking out a linear activation function and replacing it with a step function, which means now gray will only be painted onto a pixel when the incoming value is above some threshold, and we get a different pattern. They're called CPPNs because as we place hidden layers between the input and output layer, and we allow more complex cognitive architecture, we can get more complex patterns. So in essence, what CPPNs do is to compose coordinate transforms as you propagate values from the input layer to the output layer. One of the typical, what you'll notice is that if you create random CPPNs, you get non-random patterns. They tend to bias the patterns that are produced towards those that have regular gradients, like you saw in the simpler examples. In the example you can see in panel F here, there are radial symmetry, circular symmetry, bilateral symmetry. You get a lot of the regular patterns that you would like to see when you're evolving things that need to move or act in the world. Okay, so that's CPPNs in two-dimensional spaces. We can paint arbitrary patterns in arbitrarily dimensional spaces as well. So we can paint patterns in a three-dimensional space by adding an additional input neuron to our CPPN and now visit regular points distributed evenly through a three-dimensional space, feed in the X, Y, and Z coordinates, and so on. So Cheney and his colleagues back in 2014 took this CPPN concept and used it to paint not grayscale values onto pixels, but to determine whether or not to place a voxel at a given position. So we visit regular points inside this empty cage, propagate the x, y, and z coordinates through. And in this case, the first output neuron, we threshold it to a binary value. 0 means do not place a voxel at this position. 1 dictates that we should place a voxel at this position. If we do place a voxel there, we scale the second output neuron to an integer 1, 2, 3, or 4, which corresponds to red, green, cyan, or dark blue. So we're painting material properties onto individual voxels. And we then can have a third output neuron, which is going to indicate whether it's going to expand or contract, whether it's in phase or out of phase with other voxels that make up the robot. So a single cppn dictates the shape and the distribution of material properties for this soft robot. So far, so good. If there's any questions, please go ahead and drop them into chat. No problem. Okay. So I'm going to now switch and talk about computer-designed organisms. We took the approach of Cheney et al. And we adapted it for not soft robots now, but robots that are going to be constructed from biological cells. So we're going to go from simulation to reality, but reality is not going to be robots made up of metals and ceramics and electronics. They're going to be robots that are built from biological cells. So I'm going to start with simulation, and we'll talk about the biological reality in a moment. We published this work back in January in PNAS. There's a whole website dedicated to computer-designed organisms. So if you're interested in this topic, there are many more videos and images. The paper itself you can find at the website. And there's also a pointer to the source code, so you can at least you can download it and play around with simulating and evolving your own simulated computer-designed organisms. Okay, like Cheney et al., we're going to start here with an evolutionary algorithm. That evolutionary algorithm is going to generate a population of random cppns where each cppn has different connections, different activation functions inside the neurons, different synaptic weights. We're going to take each cppn in the population and allow it to construct a soft robot like you saw here. Here's one example of a random cppn creating a random computer-designed organism. You'll notice already that there are some differences between previous work and this work. You'll notice that in this case, this particular cppn painted mostly passive soft voxels, the cyan voxels. But there is a small minority of voxels here which are active. And you'll notice that they're pulsing, they're increasing and decreasing in volume at random phase offsets with one another. They all have the same frequency, the same amplitude, but different phase offsets. Why is that the case? That is because we had to build in some details here to increase the probability that we'll be able to transfer some of these simulations to reality. And I'll talk about that in a moment. So in this case, instead of 4 different kinds of voxels, there are only 2 kinds of voxels. Cyan voxels, which are passive and soft. They can be pushed and pulled by neighboring voxels. And red voxels. Any red voxels are going to have, they're all going to have the same frequency and they're going to have the same frequency and amplitude, but they're going to have random phase offsets. Here's a second random cppn in the population. We haven't done any evolution yet. We're just evaluating these random cppns. This one is made up entirely of red voxels. And again, you can see that they're increasing and decreasing in volume at random offsets from one another. Here's a third random design produced by a third random cppn. The task, like you just saw in this case, is forward locomotion, so move as far from the origin as possible. And as you can see in all these cases, the robots are not doing a very good job at doing so. So these all have very low fitness. Okay. So why are we telling evolution that the red blocks, which it can use, have these random phase offsets? The reason why is because in this case, we're going to build some of these evolved CD organisms from two types of cells. Both cell types are going to be drawn from the African clawed frog Xenopus laevis. We're going to use skin cells, which are passive and soft. They cannot move themselves, but they can be pulled and pushed by neighboring cells. And the red voxels here correspond to heart muscle tissue, or myocardiocytes. These are cells which you find in your own heart, which when placed together with other myocardiocytes, they will attach to one another and they will start to spontaneously increase and decrease in volume. If you take frog heart muscle tissue and put it together in the shape of an adult frog heart, that's familiar to those cells, and they will synchronize their action so that they all expand and contract in phase with one another, which is good news for the frog because it means that the heart acts like a pump. If you take heart muscle tissue and you rearrange it into the patterns dictated by a cppn, you have no guarantee that those cells will talk to one another and synchronize. They may not, they may partly synchronize. There are things known as chimeric states in dynamical systems theory. They may settle into a chimeric state. There's all sorts of crazy dynamics that are possible. We have no idea what they are. So we told the computer to be conservative. Assume that there's no synchronization. They will just increase and decrease in volume at random phase offsets, which makes this an extremely difficult design problem because we do not want random action at the level of the entire organism. We want these things to move straight and deterministically as quickly as possible. So the task for the evolutionary algorithm is to find a particular shape and a distribution of the passive and active material, so that together that shape and that tissue distribution will collectively de-randomize the random actions of the components of which it's built from. So just to give you a metaphor of what this is like, imagine that I asked you to design the shape of a boat. And in this boat, you're going to be able to place human rowers that are all going to row. You can place as many human rowers in the boat as you want. All of the human rowers, they will row at the same frequency and the same amplitude as each other, but they will all row at their own phase offset. What is the shape of the boat, and where should you place those human rowers? It turns out that this is actually an extremely difficult task for a human to solve, which is exactly why it's useful to use an evolutionary algorithm here. We wanted to find designs that are non-intuitive. We asked ourselves the question of, is this even possible? And because I'm here telling you about it, you can imagine that it is. Okay, so we ran this evolutionary algorithm like I just described, and one of the best designs we got back is this design, which you can see has these two large back legs. It has passive voxels on its dorsal or its upper surface, and it's patterned a bunch of heart muscle tissue on its ventral surface. And if you simulate this, even though the heart muscle tissue is acting randomly, you'll notice that you get more or less non-random action. It moves in a more or less straight line from the left side of the screen to the right side of the screen. It doesn't move very quickly. It's not very energy efficient. Obviously, we can make wheeled robots and legged humanoid robots and drones that move much faster and much more efficiently than this little creature. But speed and efficiency are not the primary goal of this project. The primary goal of this project is to see whether we can get an evolutionary algorithm to automatically design new kinds of organisms for us. Okay, that's the simulation side. Let's switch and talk about how we actually manufacture these systems now. I'm going to show you a bunch of videos here. These are taken by Doug Blackiston, the microsurgeon that we collaborate with at Tufts. You're looking through the microscope with Doug, and what you're looking at is a bunch of fertilized frog embryos. Doug is injecting some chemicals which are going to determine cell type. So at this early stage in this embryo, a lot of the cells are stem cells, which can be influenced to develop into different cell types. We're after, as I mentioned, we're after skin cells and heart muscle tissue. After that injection, after a couple of hours, Doug goes back into, looks through the microscope, and again now with micro scalpel that you can see here, micro calipers, he is gradually pulling off the vitelline membrane. It's this sort of, as you can see here, this sticky sac that holds the embryo together. He is then going to cut off what's known as the animal cap. This is several tens of thousands of cells that sit on the top, the dorsal part of the embryo. The animal cap mostly contains epithelial cells, the skin cells that we're after. So you can see him sort of cutting off this small fraction that we're going to use, and the rest of this early embryo is discarded. Just in case you're wondering, the African clawed frog produces billions and billions of embryos. So if the frog loses a few, presumably it's not too upset. Okay, now that we have the separated animal caps, which you'll see in the video here, you're going to see Doug again go back in, and he's going to try and gradually pull away the ectoderm from what he wants underneath, which is the inner cells. These inner cells are the white cells that you can see here. You can actually see the individual cells here. I said these are skin cells. These are actually at this point skin stem cells. They're stem cells, so they can still differentiate or specialize into different cell types, but they're fated to become different kinds of skin cells. Okay, so at this point he has the raw Lego bricks. He has the dissociated skin cells. He's sucked them up into a syringe, and you're going to see him now inject these dissociated skin cells back into a petri dish, and they're settling into a small concave well. So they're all settling into this location, so we can gradually pull these Lego bricks back together again, and we have pure skin cells at this point. I think this is another video of the same process here. I'm only showing you skin cells at the moment. I mentioned that we also have heart muscle tissue. So what you're watching here is a robot that's being built from just skin cells. When we need to combine skin and heart muscle tissues, Doug will lay down one layer of skin cells, and then depending on the design, he will look at the design and determine where to lay down some heart muscle tissue. Then he'll lay down a little bit more skin cells, heart muscle, skin. So it's sort of he's building up a 3D structure by layering 2D layers. So it's almost like a 3D printer. Okay, one of the amazing things that the Levin Lab has learned over the last few years, and other groups as well, is that dissociated cells, like you see here, they will basically reboot multicellularity. They don't like being on their own. They will gradually migrate together and try reconnect and recongeal into a system. So you can see the beginnings of a computer-designed organism here, otherwise known as a Xenobot. So this was our nickname for these creatures. Xeno taken from Xenopus laevis, and bot because eventually this is going to become what you could consider a robot. We also call these Xenobots because they are also alien, Xeno. Okay. So here's again a sped up version of this where you can see a much larger number of these cells pulling together. Okay, so once it's recongealed into this ball of skin cells, Doug is now going to go back in and given a target shape, which is this very simple quadruped or four-legged robot here. Given that design, he's going to use a microcauterization tool which burns away excess material. And I'll try and speed this up here. He's burning away some of this material, and you'll notice he's now making a crosswise cut in the bottom of the robot. So again, we're layering up skin and heart muscle tissue and then cutting away what we don't need. So interestingly here, the design process is automatic. We're using an evolutionary algorithm to determine where to place skin and muscle tissue. And then the manufacturing process at the moment is almost completely manual. We have a human microsurgeon who's doing this by hand. Okay, I have about 6 minutes left, so I'm going to just skip ahead. Here's a constructed Xenobot, just so you get a sense of size. So it's less than a millimeter. You can just see these Xenobots with the naked eye. Okay. Finally, in this case, Doug created this particular evolved design, and in this case, we managed to cross the reality gap. The physical Xenobot moves more or less like the simulated robot does. Clearly, there are differences between their shape and their function, the way that they move, but they definitely move. Okay, how do we know that it's moving because of the evolved design? We took the evolved design and turned it on its back, and it no longer moves. So therefore, form dictates function, and the evolved form is what's giving us the desired function, which is forward movement. Here's just some quantitative data to verify that when we simulate this creature, we get forward motion, which are the blue trajectories. If we do this a bunch of times, it always tends to move from the left to the right. The pink trajectories correspond to the movement of the simulated Xenobot. And the blue trajectories correspond to multiple built versions of this design that also always move to the left. And if we flip the virtual Xenobot on its back or the physical Xenobot on its back, in neither case does it move. So we have quantitative evidence that we can in fact cross the reality gap, at least for these simple initial Xenobots. Okay, one of the other interesting things we found is that if you built multiple versions of them and you put them in a petri dish together, they will exhibit swarm behavior. Remember that we only evolved them to move forward. So in the case of the simulated Xenobots, we know that they are not collaborating in any way, but we're not so sure about the physical Xenobots. The reason why is because cells themselves are incredibly complex machines. Cells talk to one another. Cells have their own sensor and motor systems. Maybe these Xenobots, when they come in contact with one another, the cells in the Xenobots are talking to one another, and they may actually actively be working together to connect and move around. Many years ago, Rolf Pfeiffer made Swiss robots, which was a swarm of robots that move around and collect garbage in their environment. So as sort of an homage to Rolf, we did the same thing with Xenobots. We placed these very small red pellets in a petri dish, and you can see that the swarm of Xenobots here collectively seem to be pushing these pellets into nice, neat, tidy piles. Okay. We also noticed in some cases that the Xenobots will also seem to shepherd individual pieces. This Xenobot seems interested in this pellet, even though it wasn't evolved to do so. That inspired us to go back and evolve Xenobots that could push objects. You can see that here. One potential application for Xenobots is drug transport. Here you can see a hollow Xenobot that is transporting an object. One of the nice things about Xenobots is they inherit a lot of nice properties from biological systems. One of those properties they inherit is that when they experience unanticipated situations like physical damage, they heal back up again. Uh, Another nice thing about evolutionary algorithms is we can produce lots of different kinds of xenobots. And I think in the interest of time, I'm going to skip over situating xenobots in the larger field of synthetic biology. I think you can probably understand already that xenobots are sort of a new way of combining technology and living systems. I'll conclude here. There's a pointer to the website. Future work, there's lots to do. The first one is obviously automate the way that we build Xenobots. We'd like to gain greater control over the spontaneously appearing function that we see in these Xenobots. We're currently working on adding new kinds of cell types and tissue types to our Xenobots, incorporating sense organs and new kinds of actuators. The Xenobots that you just saw have no brains. They're kind of the ultimate example of embodied cognition. They're all body and no brain, but they still do useful things. But we could, of course, incorporate nervous tissue, cells from other species. We'd like to automate sim to real. My colleague Mike Levin is interested in using Xenobots to crack the bioelectric code. How exactly do cells talk to one another? And when they do, what are they saying? We're also looking for new collaborators. If you're interested in this project, please email me or check out cdorgs.github.io. I'll finish by thanking Sam and Doug and Mike for this really exciting collaboration. And finally, thanks to DARPA here in the US for funding this work. I think I've taken all my time, but if there's time for one question, I'm happy to do so. Thank you.
Thank you. Really exciting as expected. Maybe even more exciting than expected. Before I see there is a question in the chat. I just wanted to comment that you see how this very abstract idea worked over the years, your research and other people's research, to come to something which is amazing and absolutely real, right?
Thank you, Fabio.
You see the power of deep ideas.
Thank you.
Thank you, Josh. There is a question, but I don't know if it is being made obsolete by the rest of the talk, which is, if you look in the chat, is could we use the self-reconfiguring systems to evolve creatures without the need to rely on the imperfect virtual simulation?
Yes, I actually saw that question during my talk and tried to answer it. So Lucas, if I didn't answer your question well, just let me know. But I think I already tackled that one. Again, I apologize for keeping everyone late. Maybe everyone's ready to move on. Again, please feel free to email me with questions. I'm happy to chat about this research offline if that's more helpful.
I think a quarter of an hour is actually acceptable.
Okay.
More than acceptable, and you are not guilty for that because we shifted all the previous talks. So
no problem.
Thank you again for your great talk from. So I think that now you are forced to do it all the years because people loves this idea of a deep night remote lecture.
Great. Happy to do so.
Okay. And actually, I say you don't seem to suffer too much about that. So you
science never sleeps.
Never sleeps, exactly. Okay, so thank you all for attending, and see you next year for the Shanghai lectures, and hopefully for the Shanghai conference, more or less in June. So we are a bit in the early stage, but I think it will be very, very interesting and exciting too. So thank you again. I don't know if you're going back to sleep, but okay, see you. Thank you, everybody.
Thank you, everyone. Bye-bye.
Bye. Bye.